Hello students, this is Dr. Tikia Arashiro. In this podcast, I will be talking about inorganic metabolism. Inorganic metabolism is a type of metabolism in which inorganic molecules are used by microorganisms as a source of energy and nutrients. These inorganic molecules or compounds are derivatives of nitrogen, sulfur, iron, hydrogen, and carbon and are used in energy metabolism and biosynthesis. Examples of nitrogen inorganic molecules include ammonium, ammonia, nitrate, and nitrite. Inorganic sulfurs include hydrogen sulfide, elemental sulfur, sulfate, and thiosulfate. Inorganic irons include ferrous iron and ferric iron. Inorganic hydrogen include hydrogen gas, and inorganic carbon include carbon monoxide and carbon dioxide. In general, there are three types of inorganic metabolism in which inorganic molecules are utilized by microorganisms. These include assimilative inorganic metabolism, dissimilative inorganic metabolism, and oxidative inorganic metabolism. Assimilative inorganic metabolism is a type of metabolism in which inorganic molecules are used as source of nutrients. Microbes assimilate inorganic compounds as a source of nutrient, and this process of assimilation is what you call assimilative inorganic metabolism. So let's say you prepared a culture medium containing sulfate. Now, sulfate is an inorganic sulfur and can be used as a source of nutrients for microbes. Microbes then utilize sulfate from the culture medium through a specific protein transport system called sulfate symporter. So the sulfate is transported into the cell through this transport protein, and once in the cell, sulfate is reduced to sulfide and then converted to cysteine, which is an amino acid containing sulfur. The cysteine is then used to synthesize proteins. So in assimilative inorganic metabolism, the inorganic compounds are reduced in the cell and then incorporated into organic compounds as macromolecules. Assimilative inorganic metabolism does not require oxygen, so it occurs in all aerobic, anaerobic, and facultative anaerobic microbes. Dissimilatory or dissimilative inorganic metabolism is a type of metabolism in which inorganic molecules are used as electron acceptors in anaerobic respiration. Recall that in assimilative inorganic metabolism, the inorganic molecules are used as nutrient source. So in dissimilatory or dissimilative inorganic metabolism, the inorganic molecules are used as electron acceptors in anaerobic respiration. Examples of inorganic molecules that are used as electron acceptors in anaerobic respiration include sulfate, nitrate, carbon dioxide, and ferric iron. Dissimilatory inorganic metabolism occurs in anaerobic respiration and is therefore carried out by strict anaerobic lithotrophs and facultative anaerobic lithotrophs growing in anaerobic condition. So an example that we have here are sulfate-reducing bacteria. Sulfate-reducing bacteria are strict anaerobes that use sulfate as electron acceptor for anaerobic respiration. So say here you inoculate the cells in an anaerobic culture medium containing sulfate. The sulfate-reducing bacteria will take up sulfate and reduce it to hydrogen sulfide, and once hydrogen sulfide is reduced, the cells will excrete it out of the cell. So hydrogen sulfide leaves the cell and you will observe the accumulation of sulfide in the culture medium. So in dissimilatory inorganic metabolism, the inorganic compounds are reduced and the reduced products are excreted in the environment that they are in. Dissimilative metabolism occurs in the cytoplasm. Here is the proposed pathway of dissimilatory sulfate reduction. The dotted line represents the cell wall. So sulfate is taken up by the cell through a sulfate transporter, and it is eventually reduced to hydrogen sulfide. Once reduced, it is excreted out of the cell. Here is another sulfate reduction pathway over here, and the end product is the same. It is hydrogen sulfide, which is eventually excreted out of the cell. Now, the third type of inorganic metabolism is oxidative inorganic metabolism. This is a type of metabolism in which microbes use inorganic compounds such as hydrogen gas, ammonium, elemental sulfur, hydrogen sulfide, and iron as source of electrons. So in the process, the inorganic molecules are used as electron donors. So recall that in the simulatory inorganic metabolism, inorganic molecules are used as electron acceptors. So in oxidative inorganic metabolism, the inorganic molecules are used as electron donors. Oxidative inorganic metabolism occurs mostly aerobically, although it can also occur anaerobically and carried out by anaerobic microorganisms that use inorganic compounds as source of energy. So the electron donors are oxidized, which leads to the release of energy in the form of ATP. An example of oxidative inorganic metabolism is ammonia oxidation. In ammonia oxidation, ammonia or ammonium is the electron donor. And since ammonia oxidation is an aerobic process, oxygen is the electron acceptor. So the process leads to the oxidation of ammonium to nitrite and the reduction of oxygen to water. The oxidation process requires the electron transport system. So here is the electron transport system of an ammonia oxidizing bacteria. So ammonia or ammonium enters the cell through the ammonia transport system. Once in the cell, the ammonia is oxidized to hydroxylamine by the enzyme AMO or ammonia monooxygenase. The hydroxylamine then leaves the cytoplasm and crosses the cell membrane, where it is further oxidized to nitrite by the enzyme HAO or hydroxylamine oxidase. These two oxidation steps generate protons that drive the synthesis of energy in the form of ATP. So the electrons produced enters the ATPase and drive the synthesis of ATP. Oxidative inorganic metabolism is a type of inorganic metabolism that leads to the production of energy in a form of ATP. Now I want to talk about assimilatory inorganic metabolism, particularly the assimilation of nitrate and sulfate, but I want to start off with nitrate assimilation. Now recall that in assimilative metabolism, the inorganic molecule, such as nitrate or sulfate, is used as a nutrient source. So what is nitrate assimilation? Nitrate assimilation refers to the uptake of nitrate by microorganisms. Nitrate in this case serves as a source of nutrient. The process occurs in the cytosol and is carried out not just by lithotrophs, but those that use inorganic molecules as nutrient source. Plants, fungi, algae, and many bacteria, including heterotrophs, also carry out nitrate assimilation. In nitrate assimilation, nitrate is taken up by the organism via a proton nitrate importer. Once in the cell, nitrate is sequentially reduced first from nitrite to ammonia and then into glutamate and glutamine. Glutamate and glutamine are sources of amino groups for other nitrogen-containing macromolecules in the cell. Nitrate assimilation is also the process of making ammonia from nitrate. Nitrogen is essential for all living things. It is an essential element of a living cell. Nitrogen constitutes 13% of the cell biomass, and it is a constituent of proteins, nucleic acids, and enzymes. Nitrate is assimilated by the cells to synthesize nitrogen-containing macromolecules. These nitrogen-containing macromolecules include amino acids, nucleic acids, and coenzymes. Here's an amino acid. An amino acid has an amino group, a carboxyl group, and a side chain. The amino group contains nitrogen. A nucleic acid has a phosphate group, a sugar backbone, and a nitrogenous base. The nitrogenous base contains nitrogen. So nitrogen constitutes 13% of the cell's dry mass. So let's talk about the oxidation states of nitrogen. Oxidation state or oxidation number is a number assigned to an element in chemical combination that represents the number of electrons lost or gained by an atom in a compound. It is an indicator of the degree of oxidation of an atom in a chemical compound. The increase in oxidation state of an atom is known as oxidation, and the decrease in oxidation state is known as reduction. Such reactions involve the transfer of electrons and protons. A net gain in electrons and protons is a reduction, and a net loss of electrons and protons is an oxidation. This table right here shows you the different oxidation states of key nitrogen compounds. Organic nitrogen has an oxidation state of -3. Ammonia also has an oxidation state of -3. Nitrogen gas has a 0 oxidation state. Nitrous oxide has an oxidation state of +1. Nitrogen oxide is +2. Nitrite is +3. Nitrogen dioxide is +4. And nitrate is +5. Now, by knowing the oxidation states of compounds, we can determine how many electrons are gained or lost in a reaction. Let's take a look at the nitrate assimilation. Nitrate assimilation begins with nitrate, and nitrate here is going to be assimilated by the cells as a nutrient source. So the nitrate enters the cells and gets reduced to nitrite and then to ammonia. Recall that the reduction reaction involves a gain of electrons and protons. If you were to answer the question, how many electrons must be gained by nitrate to be reduced to ammonia, you need to know the oxidation states between nitrate and ammonia and be able to calculate the difference between the two. The difference is the number of electrons that must be gained by nitrate to reduce it to ammonia. The oxidation state of nitrate is +5, and the oxidation state of ammonia is -3. So +5 minus -3 is equal to 8. Therefore, the conversion from nitrate to ammonia is an 8-electron reduction step. And this means that 8 electrons must be gained by nitrate to be reduced to ammonia. So in assimilative nitrate reduction, the net gain is 8 electrons. Also, note that the decrease in oxidation state from +5 to -3 indicates that the conversion from nitrate to ammonia is a reduction process. So let's take a look at the nitrate assimilation pathway. This pathway is present in all prokaryotes that reduce nitrate to ammonia, which is then incorporated into the cell material as cellular nitrogen or biomass nitrogen. The enzymes are found in the cytosol and are not coupled to ATP formation. So here is the cell, here is the plasma membrane, and here is the cytosol. Now, let's go back to the biochemical pathway. Here you can see that nitrate is reduced via 2 electron steps to nitrite, nitroxyl, hydroxylamine, and ammonia. The ammonia is incorporated into organic nitrogen compounds via glutamine synthase or glutamine oxoglutarate aminotransferase. Glutamate and glutamine are important sources of amino acids for other nitrogen-containing macromolecules in the cell. They serve as amino groups in amino acids, nucleic acids, and coenzymes. So the process involves 8 electrons. 2 electrons are transferred from nitrate to reduce it to nitrite. 2 electrons reduce nitrite to nitroxyl. 2 electrons reduce nitroxyl to hydroxylamine, and 2 electrons reduce hydroxylamine to ammonia. So 2 plus 2 plus 2 plus 2 equals 8. 8 electrons are needed to reduce nitrate to ammonia. The assimilatory nitrate reduction pathway involves two enzymes, the cytoplasmic nitrate reductase and the cytoplasmic nitrite reductase. The cytoplasmic nitrate reductase reduces nitrate to nitrite, and the cytoplasmic nitrite reductase reduces nitrite to ammonia. So here, nitrate is taken up by the cell via a nitrate transporter. Once in the cell, it is reduced to nitrite and then to ammonia, and then ammonia becomes incorporated into the organic nitrogen compounds in the cell, such as glutamine and glutamate. So let's move on to sulfate assimilation. Sulfate assimilation is when cells assimilate sulfur in the form of sulfate. The sulfate is first reduced to sulfide, such as hydrogen sulfide or bisulfide, and then becomes incorporated in sulfur-containing compounds in the cell. Major species of sulfides in the cytoplasm include hydrogen sulfide, sulfide, and bisulfide. Sulfur is an essential element for all life, but almost always in the form of organosulfur compounds or metal sulfides. Constitutes 1.8% of bacterial cell dry mass. Sulfur are constituents of proteins and coenzymes. Sulfur-containing macromolecules include ferredoxin, coenzyme M, methionine, and cysteine. Ferredoxin is an electron carrier in the electron transport system. It is an iron-sulfur protein that mediates electron transfer. It contains sulfur. Coenzyme M is a coenzyme required for metal transfer reactions in the metabolism of methanogens. It also contains sulfur. Now, methionine and cysteine are amino acids that contain sulfur. So sulfur is an essential element for all cell metabolism. A cell will not survive without sulfur. This table shows you the oxidation states of sulfur compounds and electron donors for sulfate reduction. Let's go over this table. So organic sulfur has an oxidation state of -2. Hydrogen sulfide also has an oxidation state of -2. Elemental sulfur has a 0 oxidation state. Thiosulfate has an oxidation state of +2. Sulfur dioxide is +4. Sulfide is +4. And sulfate is +6. So knowing the oxidation states of compounds, we can determine how many electrons are gained or lost in a reaction. Let's take a look at the sulfate assimilation. Sulfate assimilation begins with sulfate, and sulfate here is going to be assimilated by the cells or used as a nutrient source. So sulfate enters the cells and gets reduced to hydrogen sulfide. Recall that a reduction reaction involves a gain of electrons and protons. So if we want to know how many electrons must be gained by sulfate to be reduced to hydrogen sulfide, you'll have to know the oxidation states between sulfate and hydrogen sulfide. The difference in their oxidation states is the number of electrons that must be gained by sulfate for it to be reduced to ammonia. Looking at this table here, the oxidation state of sulfate is +6, and the oxidation state of hydrogen sulfide is -2. So +6 minus -6 equals +8. So the conversion from sulfate to hydrogen sulfide is an 8 electron reduction step, and this means that 8 electrons are needed to reduce sulfate to sulfide. Thus, assimilative sulfate reduction has a net gain of 8 electrons. Note that the decrease in oxidation state from +6 to -2. This decrease indicates that the conversion from sulfate to hydrogen sulfide is a reduction process. Now, Prior to the reduction of sulfate to sulfide, sulfate needs to be activated by ATP to adenosine 5-phosphosulfate or APS. This activation step is necessary because sulfate is metabolically inert. The key enzyme involved in sulfate activation is ATP sulfurylase. ATP sulfurylase catalyzes the activation of sulfate by transferring sulfate to the adenine monophosphate moiety of ATP to form adenosine 5-phosphosulfate or APS and pyrophosphate. So APS is the activated form of sulfate. It consists of an adenine monophosphate attached to a sulfur. The energy stored in the phosphoric acid, sulfuric acid, and hydride bond of the adenosine 5-phosphosulfate or APS allows sulfate to undergo further reactions. So here is the assimilative sulfate reduction pathway. The first step is sulfate activation. Recall that sulfate is metabolically inert and needs to be activated prior to the reduction process. Sulfate is activated by means of ATP via ATP sulfurylase to form adenosine phosphosulfate or APS. The second step involves a phosphorylation step by APS kinase. So APS kinase removes a phosphoryl group from ATP and phosphorylates APS to phosphoadenine 5-phosphosulfate or PAPS. So this process converts APS to PAPS. PAPS is reduced to sulfide by the enzyme PAPS reductase. Then sulfide is reduced to hydrogen sulfide by the enzyme sulfite reductase. The sulfhydrase then converts hydrogen sulfide to organic sulfur compounds. 8 electrons are needed to reduce sulfate to hydrogen sulfide. 2 electrons are reduced in the reduction of PAPS to sulfide, and 6 electrons are used in the reduction of sulfide to hydrogen sulfide. 2 plus 6 equals 8. So the assimilative sulfate reduction has a net gain of 8 electrons. That's the end of the first podcast on inorganic metabolism. Stay tuned for the second part of this podcast.